Hello, my name is Richard Michelmore, and I'm the director of the Genome Center at UC Davis. And what I'm going to do today is to describe how we've ramped up high throughput testing for COVID, and then how that led to an opportunity for high throughput characterization of variants. And really going forward, what type of surveillance strategy should we be using for monitoring for variants of concern? So just a little bit of background, Davis is at the head of the San Joaquin Valley, a very fertile area in California, about northwest of San Francisco. And what we're doing here is we're serving both the campus and the city of Davis for COVID testing. We're about 39,000 students and 1,500 faculty, and the college town is about 75,000 individuals. So that's our target population. When COVID started to ramp up the pandemic, there was a lot of discussion about the concept of high throughput testing, and much of the testing was being done on symptomatic individuals, and it was taking 5-7 days to get the results back. This really is not ideal for community-grade surveillance testing, in that many of the individuals are asymptomatic, and this window, based on data from Michael Miller at Harvard, you can see that the infectious period is only about 5 to 6 days. And if you sample at the top of that peak, you delay for a week, and then you contact trace back 2 days, it's really not valuable. So what we needed was rapid, inexpensive, high throughput workflows, and the sensitivity was somewhat secondary to frequency and turnaround time. If you want a really very sensitive test and you have slow return of results, it's really a waste of time and resources. So what we did was, I come from an ag biotech background. I knew that there was technology out there from the ag biotech sector would allow very high throughput testing. In the biotech sector, the big ag companies, they can generate millions of data points a day, orders of magnitude higher than what was being considered by the medical testing community. And the machine you see in the middle is deployed, and the biotech sectors will have multiple of these, and I say they can generate millions of data points a day. But their endpoint, we chose to go with the IntelliCube, which is the platform you see on the left, which is actually a qPCR machine that can do massively parallel qPCR reactions. And the way that they do that, if you look on the right, is there's an array tape, which is essentially an array of 768 wells plates on a tape that you can mount the reactions on and then analyze. So what we did was we started out, we were doing about 1,000 students a week as they came onto campus, 2,600 the second week, 4,500 the third week. This is really a very rapid ramp up. And since then, we've expanded to all students, faculty, staff with at least weekly testing. And, we expanded this to the city of Davis. We've got an initiative called the Healthy Davis Together, which encompasses both the campus and the city of Davis. The Healthy Davis Together is really a very holistic approach. We're just one component of that. But the rapid testing, there are kiosks around the city as well as on campus that collect the saliva samples of asymptomatic individuals. The symptomatic individuals, they go to a different collection and go through a different workflow. So we are in the Genome Center. We're a pop-up lab. We're an extension of the CLIA certified student health lab. There's an MD that's in charge of it. There are doctors obviously recommending the tests. What you see here are the two IntelliCubes here that it's very important when you set up a high throughput workflow to have redundancy. Our actual workflow looks something like this. So in the sampling kiosk, the people will spit into a barcoded tube, and that is immediately in the collection kiosk is put into a 96-well rack. From that, we can scan it, we heat inactivate, and one of the tricks to working with saliva is, as you know, saliva is very viscous, or at least some of it is, and this is problematic if you wanting to pipette. But Lutz Floenicker, who's the manager of the DNA core, tech core, had a brilliant idea of using papain. You know, when you eat papaya, your lips tingle. That's because papain is an aggressive protease that's doing a number on your saliva. And just incubating for papain can get rid of the viscosity problem. That really allowed us to work with saliva. So we're analyzing raw saliva. There's no RNA extraction. We can decap semi-automated transfer 384 well plates. Those 384 well plates are placed on the IntelliCube, and the IntelliCube does really all the rest. It sets up the reaction and runs the reaction, and then the results from the reaction are exported to the cloud, where a plugin from UsionTech uses an approach that takes into account all the curve to call positives and negatives. And if you're interested in more of this, I encourage you to watch the video. With the link is at the top. They'll walk through the process. So, question, of course, is when you're working with high throughput raw saliva, what's your sensitivity? We're running the M1 N2 FDA approved test, same primers and probes. We can get down to an LOD of about 15 copies. We're actually, the latest results, about 12 copies per microliter of saliva. And this is comparable to some of the EUAs for NP swabs. So it isn't the most sensitive test, but it's certainly sensitive enough. And with two IntelliCubes, we could do, in theory, up to easily 12,000 samples in a 12-hour day. Right. So in terms of throughput and rapid, we've now reported over 400,000 tests, and we get the results back. 33% can go back on the same day. And from this, we've detected over 1,700 positives, and because of the rapid turnaround time, we can rapidly quarantine the individuals and follow up on their close contacts. Now, the workflow is high throughput. The sample collection really isn't rate limiting. The accessioning is the rate limiting step. Even though we have a very, very simple workflow, this is what limits us. At the moment, we regularly do 4,000 to 5,000 samples. We could ramp up to 8,000 a day, I would think fairly comfortably. The two machines are not rate limiting. In many workflows, it's the analysis at the qPCR step that becomes rate limiting. But because we have this tape system, this is not rate limiting to us, and nor is the data analysis. Now, because of this, it's not being rate limiting. That gives us spare capacity to look for variants of concern and to use the same workflow, but now using different primer sets to look for variants. But before I move on to that, I want to acknowledge this has been a massive team effort. A lot of people all across campus got mobilized. We have a big team in the collection kiosks. We have three teams in the testing lab, 2 production teams and 1 development team. Data management is really important. Many people in administration never underestimate the importance of having an enthusiastic and efficient administration. It's been extremely helpful. And then all of this has been developed in collaboration with LGC that makes the IntelliCubes with perspectives from Corteva. They were very useful in the initial stages as we set up the high throughput workflow. So moving on to detecting variants. There is a real need for surveillance of consequential variants. Now, viral evolution is inevitable. Mutations are happening all the time. And most of these mutations are going to be inconsequential, but a few of them are in fact going to have a consequence. They'll increase the fitness of the virus, and because of that, they'll be selected for and increase in frequency. And the problem is there is an effectively infinite population size in the virus, continually generating mutations. And the current selection is for increased transmissibility, and that's what we've seen. You get the B117s, the B13.1s, the P1, etc., that originally were identified in the UK, Africa, and in Brazil, respectively. So as you get a rollout of vaccines, that puts a very steep selection pressure that will render the vaccines less effective. So increased virulence and vaccine escapes are probably inevitable. Some predict that vaccines will not even last 12 months. What we need to do is to try and minimize what we call a boom and bust cycle. I'll have a slide on that in the next slide. And I think the scary thing is, what we're currently doing is only practice for when vaccine variants appear that are not controlled. So if anyone has any doubts about whether evolution can occur, they just need to have a look at what's happening with SARS-CoV-2. So what I mean by the boom and bust cycle, I say I come from a plant disease background. With plants and plant diseases, there are recurring plant disease pandemics. And actually wheat rust that devastated wheat production in the US in the 50s, you get this term where you screen germplasm for resistance, you identify a source of resistance, you breed it, release it, it increases in acreage, steep selection pressure, pathogen changes, you're back to a susceptible cultivar. And you go round and round this cycle, boom and bust cycle. And this is what's likely to happen with vaccines. We'll have highly effective vaccines deployed, and that puts a steep selection pressure on the pathogen, the virus. Changes will occur that render it ineffective. We'll need to develop more vaccines. What we need to do is to try and slow down this cycle. We need to maximize the evolutionary hurdle for the pathogen. And the key to that is monitoring the pathogen. So, SARS-CoV-2 will continually evolve, though it's actually evolving relatively slowly to some other things like the flu virus. There are 3 new strains of current concern: B117, B1351, and BP1. They may be more contagious. There's a particular mutation known as N501Y, and another one, the E484K, which is in the binding site. What's shown here is a model of the structure of the SARS S protein, which is binding the interface to the receptor. And you can see these mutations are on the receptor binding interface. It was initially identified actually because one of the primers for a commonly used test for COVID was actually spanning a deletion for one of the mutations that is present in the B117 strain. Our test is a very common test for the M1 and M2 that hasn't yet been affected by mutations in the virus, but we're certainly looking out for that. So what we now do is use our technology with known mutations to monitor for variants of concern. We're also monitoring wastewater. This is a different project. And wastewater is turning out to be very, very informative as to whether you've got the virus there. Why we're interested in this, clearly some of the viruses, some of the variants are apparently less controlled by the vaccines already. The data is still coming in as to which vaccines are more or less affected by these new variants coming in, and this is really beyond the scope of this current talk to cover all of that. But suffice it to say, it's extremely important to monitor for variants. The question is, how do we do that? I think the two things on the table are whether we genotype or whether we sequence. Sequencing is obviously the gold standard. You can sequence the whole virus, you can look at it, you can look at the phylogeny, but there's a cost. You really cannot. One of the biggest things about sequencing is the throughput is so much lower. The cost is higher. You have to extract RNA. It's relatively, the complexity is higher. The turnaround is certainly much slower. As I said, the resolution is much better. And of course, if you're only doing genotyping for known SNPs, you won't get de novo discovery. So I think we need to concern, why are we doing what? The two technologies are complementary. If you do SNP genotyping, so you're just looking at positions in the genome that you know could be consequential, you can then monitor in high throughput for variants of concern. You won't detect new consequential variants, and that's why we certainly need to be sequencing as many isolates as possible and getting a good random sample. The genotyping, the message here, I think, is genotyping is complementary to sequencing. So the type of genotyping we do, and again, I'd say we're developing these SNP assays in collaboration with LGC. The IntelliCube and the Nexar equipment that I showed you in my third slide, they were actually developed to monitor for SNPs. They were designed, and therefore it's the ideal technology to deploy for high throughput SNP analysis. And the way it works is you have a forward and reverse primer, and this is just the same assay as been used for detecting the virus in the first place, except in this case, you have not only a probe, a match probe, but you have a mismatch probe. And the way that these assays work is you have a short RNA sequence that is targets over the polymorphic position, and these sequences these probes are not fluorescent because they have a quencher that quenches the fluorescent signal. Now, if you have different fluorochromes linked to different match and mismatch probes, they will compete for each other depending on the sequence, and you will get a strong signal when there's a match and a weak signal when there's a mismatch. So we have up to 14 assays currently working. For those familiar in the field, will be very familiar. They're the usual suspects of sites that we're targeting. And you can see the variants of concern here and a variant of interest. And you get a different pattern of presence or absence of the variant site, which will allow us to rapidly distinguish between the different strains. The type of readout, here's a 2-PCR trace off the IntelliCube. You can see that here's all the wild type ones going on in blue off to the left. And actually, we're running samples here. These are either raw saliva samples, or in some cases, they were actually from symptomatic individuals where we've taken the leftover from the Panther run. So this is the nasal swab in VTM. And you can see we get a really nice signal there too. And then you've got the variants running along the bottom. So you can see very clear distinction between the fluorochromes coming off, the fluorescent signal coming off from the variant as from the wild type. So this is the deletion that's characteristic of B117. Here's the N501Y. You can see we've got, again, several B117s here. Up the wild type, clear distinction. Then E484K, this is not present in the B117, but it is present in P1 and the B1351. And here we've got clear distinction from the variant strain from the wild type. So here's some examples of different SNP assays. What we're showing here is the endpoint analysis, just a little bit easier to see for eight genotyping assays. And you can see we get really good clustering of the variants from the wild type, which allows us to call fingerprint each sample. The CT value, the cutoff is about a CT value of 30, which is about the same threshold for sequencing. Below 30, we get strong signal. 30 to 32, we're getting more and more dropouts. And above 32, 33, we really don't get any reliable signal. So what we're currently doing at Davis, we can take a variety of inputs, raw saliva, swab samples, or extracted RNA. We're screening all our positives. So all the positives that come through from the asymptomatic screening, and then we're also picking up symptomatic individuals as well. We genotype them 14 loci. We have rapid turnaround. We can have informative data with a few samples of collection. You know, and all the non-wild type samples, the variants of concern, variants of interest, or things that just don't conform to the haplotype that we'd expect, we sequence validate them. The Illumina sequencing takes another week or so, and then we report the results to the Yolo County Health Officer. So an example of this, we had a sample, and it was collected 26th of March. 2 days later, we genotyped it, and the genotype, as you can see here, was variant for E484K, K417N, and the N501Y. Now that is characteristic of the B1351. You can see it matches this very nicely, as it did for the other loci. Send that off for sequencing, and about a week later, we had it sequence validated. So doing 100% of our positives get genotyped, and then it allows us to pick up the frequency of each of the variants. So, summarizing, the SNP assays of variants, the advantages are they're high throughput, they're rapid, they're inexpensive, and there are multiple types of SNP assays available. We're not the only show in town. Several different groups are rolling out SNP assays. Anything that's basically PCRable, you can use as a template. In many cases, you can use the residuals from whatever test you're running in whatever primary test you're using. There is no need for RNA extraction. You have a very flexible and adaptable panel of informative SNPs. As the sequencing discovers more SNPs, variants of concern, and the critical SNPs that are characteristic of that variant, it takes about hopefully a 3 to 4 week turnaround time from the discovery of that to deploying it commercially. It can be run on any qPCR machine. And in fact, it's very low tech. It could be just endpoint assays. Anyone with a PCR machine and a fluorescent plate reader could be generating this data. Now, a thing to stress, it's very definitely complementary to sequencing. We can monitor very large numbers of samples. the information generated in real time to allow public health interventions. But it doesn't replace the need for sequencing. It stands on the shoulders of sequencing to identify new SNPs of interest. But it does allow triaging samples to be sequenced and reducing redundant sequencing. If you think about it, the novel information content of sequencing is reduced asymptotically as more and more sequences of more and more samples are sequenced. Also, it doesn't compete with the supply chain for sequencing. Now, I think the technical challenges are minimal to doing this. What is much bigger are the regulatory challenges. Now, this is not an FDA authorized test, and it's ambiguous to what the point is still being worked out as to what extent that we can report the genotyping results, or we have to wait for sequence validation before reporting them. And I think this is very important, that the regulatory environment has to change so we can fully exploit the potential of this technology. The importance of testing and surveillance after vaccination. You know, the vaccines, they're being rolled out in impressive speed, but the efficacy data relies on the prevention of disease. How many people became symptomatic? Nucleic acid tests were not part of that efficacy calculation. And we know, though, that 40% of infections are asymptomatic. There's little data, though it's beginning to come, on how frequently vaccinated individuals become infected and are infectious to others. So it's critically important to monitor for variants of SARS-CoV-2 that reduce the protection of the vaccines. As we've already mentioned, B1351, identified in South Africa, has many changes in the spike protein, and it's been reported that the vaccines are not so effective against it. And as I've said earlier, the evolution of variants that compromise the effectiveness of vaccines is probably inevitable. Therefore, it's very important to continue testing after vaccination. Something that we've experienced is as the frequency of vaccinated individuals go up, people say, I'm vaccinated, I don't need to get tested. I think that's heading for a train wreck. We really need to make sure that vaccinated individuals do continue to get tested. So this requires monitoring and genotyping of results in the context of vaccination records. And as soon as we see a vaccine escape, of course, that gets plugged into the sequencing pipeline. So, what are the future prospects? Genotyping is rapid, inexpensive, and you can do a large proportion of positives. The technology exists where you could actually genotype the majority of positives detected in the US. It's just a matter of the will to deploy the technology to do this. What's also very important is that there should be surveillance on a global scale. And in many countries around the world, sequencing is just not feasible. But these relatively low-tech tests could be performed. And then the alarm could be raised when you get variants blowing up in certain areas. Obviously, we still need to sequence from these areas. But intense sequencing from much of the world is just not feasible. Again, stressing it's complementary to sequencing. What we need is continual development and deployment of new genotyping assays with a 3 to 4 week development cycle. We need regulatory acceptance for public health actions. And ultimately, I think we should be work towards getting FDA approval for genotyping by CLIA labs for patient interventions. So when strains that clearly are overcoming vaccines, that then is actionable at the patient level. And right now, the sequencing is way too slow. So we need to work out a way of rapid genotyping to say which variant a patient has and then drive the intervention. So that's it. I'd like to wrap up. And then the credits here, this has been a lot of work from the testing and sequencing to work out the assays. Particularly, I'd like to credit the LGC design team, Lindy McLennan and Tanya Nolan in particular, that have helped us. They give us the probe primers and probe sets. We test them out on experimental and clinical samples. It's been a tremendous interaction. And with that, I will wrap up. Thank you for your attention.